Today we are going to talk about the primary structure of proteins or polypeptides, and how the primary structure or sequence of polypeptide leads to various types of secondary structure. Here we have the basic unit of a protein or polypeptide, the amino acid. All amino acids contain an alpha carbon to which is bonded covalently an amine group, a carboxyl group, a hydrogen, and various side chains or R groups. The R groups come in many different sizes and structures, from glycine, which has only a hydrogen, to phenylalanine with its large aromatic structure, to glutamine, arginine, and glutamate, which are polar and either uncharged, positively charged, or negatively charged respectively. In the process of building polypeptides, the carboxyl group of one amino acid undergoes a condensation reaction with the amine group of another, forming a dipeptide and water. The resulting bond is called a peptide bond, due to resonance between the carbonyl carbon and the amine nitrogen. A peptide bond is planar. This is often depicted as a double bond between nitrogen and the carbonyl carbon. Over 99% of peptide bonds are formed in the trans configuration, with the exception of the amino acid proline, which prefers the cis configuration much more frequently. The sequence of a polypeptide determines the secondary structure of that section of the protein. Because a peptide bond is planar, the polypeptide can only rotate along the bond between the nitrogen and the alpha carbon. This rotation angle is designated as phi, and along the bond between the alpha carbon and the carbonyl carbon. This rotation angle is designated as psi. The rotation angle between the nitrogen and the carbonyl carbon is designated as omega, but is rarely significant, as it is almost always the same. These dihedral angles phi, psi, and omega are described as being either positive or negative 180 degrees at the most extended, or 0 at the most sterically compressed. Negative degrees are found on the right of the bond, if you are looking down the barrel. Positive degrees are found on the left, plus or minus 180 degrees, negative 120 degrees, negative 60 degrees, 0, positive 60, positive 120, positive 180. There are three typical secondary structures: alpha helices, beta sheets, and beta turns. An alpha helix tends to form when phi and psi angles are able to both reach negative 60 degrees. This is the preferred structure, as the amine of each residue is able to participate in hydrogen bonding with the oxygen of the carbonyl carbon 4 units away from it. Also, R groups form on the outer face of the helix. However, steric interference and polar crowding can make 120-degree turns to the right unlikely. In this situation, a beta sheet may be preferred. In a beta sheet, phi angles are positioned at negative 120 degrees, while psi angles are positioned at positive 120 degrees. This creates a wave-like structure with R groups poking up and down from the beta strands. Beta strands can then interact through hydrogen bonding with adjacent strands in either the parallel or anti-parallel configuration. Anti-parallel beta sheets are more stable due to the preferred angle of hydrogen bonding, 180 degrees. The third type of secondary structure is a beta turn. Beta turns are generally caused by the amino acids proline and glycine. Since the R group of glycine is just a hydrogen, glycine is the least restricted amino acid structurally. Proline, on the other hand, is extremely restricted, as its nitrogen is bonded to both the alpha carbon and its R group, often resulting in a cis peptide bond. Now, let's put this all together. This is a model of a tripeptide, which consists of alanine, glycine, and proline, going from the N-terminus on the left to the C-terminus on the right. Amine groups are found to the left or upstream of each alpha carbon. Carbonyl and carboxyl groups are found to the right or downstream of each alpha carbon. R groups are also attached to the alpha carbon and, alternatively, come into and out of the plane of the screen. In a tripeptide, there are two peptide bonds. As mentioned earlier, these bonds are planar and most often in the trans configuration. This tripeptide contains a proline bonded in the cis configuration, which is one of the main causes of beta turns. Phi angles are located between the nitrogens and the alpha carbon, and psi angles are found between the alpha carbon and the carbonyl carbon.